Great, okay, so as evidenced by that herp quiz, I'm pretty new to the world of herpetology. I just moved here to Fort Collins last March, and this is my first project working with herps, and it's been very exciting and super awesome. So today I'll be talking about the work I did this past season that Boyd touched on with Colorado Parks and Wildlife. And the title of this presentation is Investigating Factors Affecting the Distribution of the Northern Leopard Frog, and it's focused in northeastern Colorado. Here's just a quick overview of what I'll be talking about. Just a quick outline of the presentation, but I'm going to jump ahead since I have 10 minutes. And just jump right into the background on northern leopard frogs. So northern leopard frogs, as you can see by this native range map, are very broadly distributed across North America. So they go all the way from the East Coast almost to the West Coast, and they've got like a lot of different habitats that they occupy too. So they're very like general species. However, despite this very broad geographic range, there have been a lot of documented localized declines. And this corresponds with like a lot of what we see in the literature, where in the 70s and 80s, we started seeing pretty bad declines of northern leopard frogs. And this is focused in the Intermountain West. Additionally, in 2011, there was a study done by Peter Johnson, and this study was done in just the state of Colorado, and it looked at northern leopard frog occupancy in the whole state. And out of all regions of the state, it found that in northeastern Colorado, northern leopard frogs were declining most severely, and it was significantly worse than all other regions of the state. They are also listed as a Tier 1 species of greatest conservation need in Colorado. And some of the factors that have been contributed to their decline, as similar to other amphibians, are habitat loss and destruction. We see a lot of development along the Front Range and in northeastern Colorado, so that could be a contributing factor, as well as invasive species like the American bullfrog, which we heard about earlier. And these guys are pretty bad predators. They occupy habitats in great numbers, and they kind of take over. And finally, we talked a lot about BD, which is the causative agent of chytrid fungus. So this pathogenic fungi has been really destructive to amphibian populations. And despite the fact that we know some of these causes, we still don't know a lot about them in detail. And so the information needs we still have out there include basic distribution information on northern leopard frogs, as well as the role of BD. So BD has been studied in the mountainous regions a lot, but hasn't been studied in northern leopard frogs very extensively. And like you can see, they occupy a lot of different habitats, so they're in a lot of different places too. And finally, the role and control of bullfrogs hasn't been explored in great detail with northern leopard frogs, and so understanding more about those dynamics is really helpful. Finally, this research is really well timed, as there's currently this grassroots movement that Boyd talked about, the Northern Leopard Frog Working Group, as well as just a lot of other community members and scientists that are getting really interested in northern leopard frog conservation. This brings me to my research question, which is what are the relative impacts of habitat alteration and invasive species, those being BD and the American bullfrog, in determining change in northern leopard frog distribution over the past decade. So that study in 2011 did a pretty good job of covering a lot of different areas in the state, but since then we haven't really looked at northern leopard frog distribution in great detail. And I will say that this is one of a few research questions that this study is looking at, but it's the one I'm going to focus on for this presentation for the matter of time. So this brings me to our sampling design. We confined our study to the South Platte River Basin, and we confined it to the Colorado portion. That's Boyd's area, and he was one of the leading members of this study, so that's where we looked for frogs. And this map on the left shows all of the HUC-12 boundaries that we have northern leopard frog data for. You might be wondering, well, what is a HUC-12 and why do I care about that? But it's basically just a way we spatially classified how we were going to do our data collection. And it's a classification system for delineating watershed boundaries. That 12 number after it is just a unique identifier code that specifies the spatial scale of that watershed. So 12 is smaller than a HUC-9. And we just thought HUC-12s were the appropriate scale for this study and what we wanted to accomplish. This map has a couple of different colors on it. So all the grayed out area are the HUCs that we don't have any data for northern leopard frogs for. And I will just pause here to shout out to all the people that contributed data to this project. including CNHP. They were one of the big contributors. We also did use some INAT and HERT mapper data, and there were just a bunch of different contributors to the data portion, which was really helpful. So this orange color shows hucks that we have historic data on northern leopard frog occupancy, but we don't have data in recent decades. The yellow color shows recent data, but not historic. So northern leopard frogs primarily were identified with iNaturalist observations in these hucks, and they hadn't been identified in the past. The red colors show our hucks that we have pretty good data for northern leopard frogs. So this would be the most recent decade and 2 other decades. So those have the best chance of finding northern leopard frogs. This map on the right just shows the random subset that we drew for our sampling season. So unfortunately, we couldn't go to every single place we wanted to, so we just had to randomly draw where we were actually going to go on the ground and look for frogs. And that just shows you the difference in the area that we have all our data for and the area we actually went to. So you might be wondering, where did we actually go look for these frogs? Each of these hucks are actually quite big, and within each huck, we chose 3 sites to go look for frogs. And we sampled those sites based on the quality and quantity of data we had. So for instance, if we had a site that had like 3 different observations of northern leopard frogs versus a site that just had 1, we would prioritize that, as well as breeding data. So if there was data on like larval stage amphibian, then we would choose that over non-breeding data. And we classified these sites based on the type of habitat. So we called them ponds, reservoirs, wetlands, and streams. And we also took down a lot of other habitat data, but these are just our 4 main categories for our sites. We surveyed every site 2 times over our field season for northern leopard frogs and bullfrogs, as those are our primary amphibian species of interest. And we incorporated both the northern leopard frog breeding season and the bullfrog breeding season. So our field season was April to October, because bullfrogs breed a little later than northern leopard frogs. The field methods we used, we had a couple different field methods. So at every single site, we did visual encounter surveys, and those just basically are exactly what they sound like. We just walked around, we looked for frogs, and if we visually encountered them, we wrote down what they were, where they were, and some more information about that. We did those 2 times over our field season at every site. And we also installed these automated acoustic recorders, or song meter minis, and that's shown in the picture with that little box on the pole. And we put these up early in the season, and we left them continually recording throughout the season. And they're just recording the breeding calls of northern leopard frogs and bullfrogs, as well as a lot of geese. And then I got to go through and listen to them and figure out what species we had. And then we also did environmental DNA collection. So that backpack contraption is what we used to collect our environmental DNA. And we did our acoustic recorders and our environmental DNA at a subset of sites, which I could touch on later, but basically it's part of a methods comparison portion of this project. So we just did those at a subset and not every single site. And then finally, we have this component of BD swabbing. We really want to get this updated distribution of BD in the South Platte River Basin. And to do that, we need to know where amphibians are. So in 2022, it was a lot of looking for amphibians and trying to find them. And in 2023, we're going to go and swab a bunch more sites. But we did get a couple sites swabbed, and we got more sites swabbed with the help of Boulder Open Space and mountain parks. And so we're going to go back and get some more samples in 2023 and hopefully get that information out soon. This brings me to some of our preliminary observations. So I haven't analyzed any of this data yet, but we have looked at it, and you can see pretty clearly just some visual observations from the data that I personally find quite interesting. So this map shows those sampled hucks that we drew for 2022 and went to and surveyed. And the blue color shows places where we only saw bullfrogs in these hucks. We did not see any northern leopard frogs. Green shows just northern leopard frogs, and that orange shows both species, and then our gray shows neither. And one of the interesting takeaways, I think, is that these blue colors show our bullfrogs concentrated in a lot of these urban areas. And urban areas have a lot of like retention ponds and reservoirs and altered bodies of water that have become permanent, which really benefits bullfrogs. This is because their tadpoles have to overwinter before they turn into frogs, and so they really rely on these permanent bodies of water. Whereas northern leopard frogs, they can survive in a lot more ephemeral. And then you see both species, as well as northern leopard frogs, kind of spread out throughout. And that's cool because northern leopard frogs do have very broad geographic range, and we're still seeing them exist in a lot of different habitats. So that's pretty neat. But one of the things that I find interesting as well is the gray color with neither species. All of our mountainous sites had neither species, and these high elevation sites in particular really didn't have any northern leopard frogs or any amphibians aside from an occasional tiger salamander. And that kind of corresponds with like BD data showing a lot of these high elevation sites having localized extinction events, as well as climate change potentially impacting high elevation sites more severely. So these are just some preliminary observations, but kind of interesting. And I'll talk about my other research objectives because they're a big component of this study, but I didn't have time to dive into the detail on every single one of them, so I'll just outline them for you here. We do want to test the efficacy of our 3 detection methods, those being visual encounter surveys, our automated acoustic recorders, and eDNA collection. And what we want to do is just see how good each method is at detecting our species of interest and what circumstances lead to better detection. And ideally come up with a monitoring framework where people could use these methods in a very complementary way in the future and use them more cost effectively as well. We also want to look into these habitat characteristics that might be promoting American bullfrog colonization events or allowing northern leopard frogs to coexist with bullfrog populations. And that second point I find particularly interesting because we don't really know if northern leopard frogs can successfully coexist with bullfrogs. And there's been a lot in the literature showing that their populations do decline, but if they've coexisted for a while, there might be reasons that they could continue to do that. So those are other research questions. And this is a massive collaborative study that had so many people helping out. This list is just a small subset of all the people involved. But a big thank you to everybody who has put in so much work and time and effort on this study. It's been awesome. And I'll take any questions. Yeah.
In the hucks where you found the 2 species, the ones you had colored orange, did you oftentimes find them in the same ponds? Or were they in the same huck but in different ponds?
Yeah, so the question was, in the hucks where we found both species, did we find them together at the same site or at separate sites in the same huck? And the answer to that is both. We did have a couple sites where we found northern leopard frogs and bullfrogs in the same exact habitat, like, you know, a couple feet away from each other. But in some of those hucks, we found them at separate sites, maybe somewhat close by, maybe not super close by. And it kind of varied the hucks that they did co-occur at the exact same site. Sometimes there'd be like 2 northern leopard frogs and a zillion bullfrogs, but we had one site where we had very good population numbers of both, including a lot of juvenile northern leopard frogs. So that site particularly is interesting because they could be successfully breeding there along with bullfrogs. So one of the questions we want to look at is how habitat could play a role there. Yeah.
Have you been able to look into anything for like automating the filtering of that acoustic data?
Yeah, that's a great question. I spent a lot of hours trying to figure that out. And it is kind of challenging, but basically, yeah, you can automate the filtering of that data. And really, it just depends on how successful the program is at doing that. And it is good at detecting northern leopard frogs and bullfrogs. But if you want to parse out like phenology data, trying to figure out when they start calling and when they might end calling, then it gets a little trickier to pull out every single call. So varying success, but I'm trying to train it to be better. Yeah. Yeah.
Is there a way to relate the eDNA to like the population size? Is that a thing?
Not that I'm aware of.
More of like a presence absence?
Yeah, we're primarily looking at like, are they occupying a site or did we not detect them? So detection or non-detection data. Yeah.
At the high elevation sites where you didn't find anything, did you expect to find chorus frogs?
We did find actually some chorus frogs, not super common, but yeah, those were species that were pretty spread out. I forgot about those. In addition to, we saw tiger salamanders, but I think we did see a chorus frog or 2 as well. Definitely at one of our sites in Netherlands, we saw chorus frogs. So they're up high. And we want to hopefully swap those sites to get BD results in 2023. So.
Thank you, Waverly.